Hello all, welcome to my channel. In this video we are going to discuss about life cycle of Rhizopus. It is commonly known as bread mold and it is a saprophytic fungus. As you see in the display image, it is grow on the surface of a bread, hence called bread mold and also a saprophytic fungus. So, let us discuss details about the systematic position. Phylum is Zygomycota, class Zygomycetes, order Mucorales, followed by family Mucoraceae, genus is Rhizopus, and the species is Stolonifer. So, let us discuss about the morphology. As you see, it is dark grayish brown in color normally. The size of a sporangia is 0.2 millimeter and the size of rhizoid is 10 millimeter in height. Here is the rhizoid and it is mostly branching body. As you see, here is the branch. So, let us discuss in details. They are fast growing fungi and have a cottony appearance. Body of Rhizopus consists of branched mycelium. Mycelium is synocytic. Synocytic means there is no septation in the mycelia, and hypha is differentiated into stolon. And stolon is present in the internodal region. It is aerial and touch the substratum in the nodal region. Here is the nodal portion where rhizoids are developed. Here also the nodal portion. Next is rhizoid. Rhizoids are formed where the stolon touch the substratum. Where stolon touch the substratum, rhizoids are developed. Obviously, rhizoids have the function for anchoring the mycelium on the substratum and absorb the food material. Okay. Then next is sporangiophore. Sporangiophore are the aerial reproductive mycelia and consist of a sporangiospore terminally. Here is a sporangium inside which these are the sporangiospore. Here is the sporangiospore. We all know fungal cell wall is made up of chitin. So, as Rhizopus is a fungus, the cell wall is made up of chitin. Next is reproduction. Rhizopus reproduce both vegetatively, sexually and asexually. Vegetative reproduction takes place by means of fragmentation and each of the fragment of a stolon develops separately to form a new mycelia. And asexual reproduction takes place by means of a formation of a sporangiospore and chlamydospore. So, let us discuss about the formation of sporangiospore. Generally, sporangiospores are formed during favorable condition. Sporangiospores are non-motile spores and also called aplanospore. As the name is sporangiospore, it develops on sporangiophore. That's why the name is sporangiospore. Sporangiospore develop in a tuft from the upper side of a node opposite to rhizoidal hypha. Suppose this is the here is the rhizoid, this is the stolon and this is the rhizoid. Sporangiospore is developed just opposite to the rhizoid. Okay, and the nuclei and cytoplasm push more towards the apical side. Consequently, the apex is swollen up. As the nucleus and the cytoplasm moves towards the apical portion, here is the sporangiophore, and here is the nucleus and cytoplasm gets inward, and as the nucleus and cytoplasm are there, it starts to swell up. Swollen part enlarge and develop into a large round structure. With maturity, the protoplast inside the sporangium is differentiated into thick dense layer, and this thick dense layer is known as sporoplasm here. Thick dense layer is known as sporoplasm, and the sporoplasm is cleaved into two. The lower part is form a U-shaped columella. It is called as a columellaplasm or cleaving sporoplasm. A series of small vacuolar layers, small vacuolar structures are formed inside the sporoplasm. Here you say sporangiospore inside the sporangium. It is vacuolated structure. And these vacuolated sporangiospore rupture the wall of a sporangium and comes out. And under suitable condition, it germinates to form another mycelia. Okay. Next is chlamydospore. Formation of chlamydospore. Sporangia are formed under favorable condition, but chlamydospores are formed during unfavorable condition. During unfavorable condition, thick-walled nutrient-rich intercalary mycelium segmented by septation. As we know, Rhizopus are synocytic, means it doesn't show any septation, any partition in the mycelia, but under unfavorable condition it shows septation. Here is the structure of a mycelia, shows septa. Here is the septation, partition wall, and these partition septate wall are detached from the mother body. Here is the chlamydospore detachment, this green color structure, and a thick cell wall is formed inside, which contain protoplasm, and under favorable condition, as it formed during unfavorable condition, when favorable condition back, these chlamydospore germinate to form a new mycelia. Here is the structure of how chlamydospore form into a mycelia. I hope you have the idea about chlamydospore. Next is sexual reproduction. Sexual reproduction takes place by fusion of two compatible hypha. Most of Rhizopus species are heterothallic, having different mycelium. Mainly there is the two different mycelia mentioned as negative and positive strain. Here is the two hypha, two compatible hypha. One is from positive strain, another is from negative strain. As it is opposite, it is comes in contact, and the tip of sporangia, the tip of this strain are ruptured, and the cytoplasm and other cellular material are transferred to the apical portion. Here is the deposition of this central part, and as both the progametangia are fused to form gametangia at the apex, and rest non-cellular part is known as a suspensor. Gradually, this gametangia developed into a zygospore, and later zygospore germinate to form a new mycelium. Zygospore develop a sporangium as in sporangiospore, and sporangium is the spore bearing organ. Sporangium rupture and the spores are comes out during favorable condition. It germinated to form a new mycelia. Here we finish with structure and reproduction, overall the life cycle of Rhizopus. You have get the notes in your description box. So goes through this, and if you have any query, please comment in the comment box.